Today is our third session for the chapter that is molecular basis of inheritance. And in the last session, we studied about the replication steps. Yes and no? See here. So, yes, we know very well that is central dogma of molecular biology. And today we will learn about the transcription, that is how DNA converted into RNA, or simply it is called mRNA, right? That steps is called transcriptions. So in this entire session we will learn that what is transcriptions, and also we will little bit more about that gene, what are the genetic label of gene, their structures and the nature. We will also learn about the RNA, that is ribonucleic acid, and their types. So yes, let's begin with this. So yes, now recall that means of this is what in the last session we studied about, that is replication process. That is the initial stage of molecular basis of inheritance, where the DNA is going to multiply in number, that means of copy, right? So that is replications. We have already studied in the last session. Okay, now one beautiful image in front of you. Now tell me what that image represent. Yes. Come on, tell me what this image represent. No idea. Why you all silent? Is there any problem? No? Yes? Come on, tell me. Yes? Okay. So Anmol, they said reflection. Okay. So that is in case of physics, but we are here studying biology. So reflection, actually what this image represent, actually transcribed, right? That means of complementary, not opposite. It's a complementary to the original stand, right? So yes, we are going to learn here that how exactly that gene, at the replication stage, how it form the real image, right? So here actually it is called the transcriptions, that how the actual image is going to reflect on that particular mirror, right? So similarly we can correlate this related to the transcriptions. Let's begin with the first gene. That what is gene, because gene is important, and because you know that what is gene. Gene is a nothing but it is a particular segment of DNA, right? You have already studied the structure of DNA in the last session, right? So yes, let's talk about the DNA. So when we're talking about the DNA, yes, you have already studied the basic structure of DNA and all. But in case of gene, when we're talking about the gene, this term was first time introduced by Johansen in the year of 1909. Okay, so it was Johansen who first time introduced this term. Let me write here. Is that pen is visible to all of you? Yes. So Johansen, who first time introduced this term, that is gene, in the year of 1909, right? So prior to this man, Mendel had used the word factors. You all know about that in the chapter principle of molecular basis of inheritance and variation. We have already studied that Mendel, before this Johansen, he used that term that is called the factor for the specific, distinct or particular unit of inheritance. that take part in the expression of the trait. That means of gene is what? Gene that going to express the traits, right? So Johansson has defined gene as an elementary unit of inheritance which can be assigned to a particular trait. That means of one gene is a kind of elementary unit, okay, of inheritance which has assigned a particular trait, means they are going to represent by this particular trait. So Morgan works suggested that the gene to be a shortest segment of the chromosome which can be separated through the crossing over and can undergo mutations, influence, expression or more trait, right? So in presently, how we can define gene? So in presently we can say that a gene is a unit of inheritance which composed of a segment of DNA, or we can say the chromosome. Either it can be a segment of DNA or the chromosome situated at a specific locus, right? Like suppose in the DNA, this is the DNA double stranded. So at the specific location, okay, so at is specific locus which carry coded information, means they have something that represent the traits. That means that the coded informations associated with the specific functions and can undergo a crossing over as well as mutation. That means of that particular gene can undergo a crossing over as well as mutations. So these are the capability of gene, and we can express gene in this way, right? So this is what actually gene. What is gene? So gene is defined as a unit of inheritance which composed of segment of DNA or the chromosome situated at a specific locus which carry coded information associated with the specific functions and can undergo crossing over as well as the mutations. Now this gene, as I told you, this is a functional unit, right? Gene is a functional unit of what? Inheritance. And it is the DNA sequence coding for an RNA. That means of this gene is the DNA sequence coding for RNA, means RNA, if suppose there is RNA, mRNA, rRNA or tRNA, that is that DNA having that coding sequence of RNA. And inside that, citron is a segment of DNA coding for polypeptide during the protein synthesis. Like you can see here that this is the particular cell of any living organisms, and you know very well that inside there is a nucleus, and nucleus having a chromosome, and this chromosome, the arms of the chromosome, spirally coiled with the bands or the thread like structure that is called DNA, and the particular segment of the DNA is called the gene, as you can see here, right? So what is cistron? Cistron is a segment of DNA coding for polypeptide during the protein synthesis, right? It is the largest element of a gene. Now, what are the type of gene? So basically, when we talk on the structural gene, especially there are structural gene and promoters and all. So structural gene is specifically in the transcription unit is of two types. The first one, that is called the monocistronic structural gene. Monocistronic structural gene. And the another one is called as a polycistronic structural gene. Now, what is the difference between this monocistronic and the polycistronic structural gene? So as you can see here, the monocistronic or the polycistronic gene, that it is seen in eukaryote. Monocistronic structural gene is also called the split gene, which is basically seen in eukaryote. Okay, so all eukaryotic organism you will get a monocistronic structural gene. That means of here the coding sequence exons are interrupted by introns, and that is called the non-coding sequence. So exon appear in the process of mRNA and intron do not appear in the process of mRNA. That means of in that monocistronic gene, when you see the actual structure of this monocistron, it is like, this way. Suppose this is the segment of gene, and on this segment there is alternate order of exon and intron, like this. So this segment, shaded segment, I can say, okay, this I can call it as a coding this I can call it as, this is the exon, and this one is introns. This is again exon, this is exon and intron. So what happened, this intron is not required for the process of transcriptions, so that's why the removal of this intron is taking place, especially in eukaryote. And there is a requirement of coding sequence which is called exon. So this exon and this exons are going to be joined together by DNA ligase. So so this exon, this removal is called as a spliceosomes. Okay, that we will see what are the process in the next slides. Okay, so generally you have to remember that is in the monocistronic there is a coding sequence and non-coding sequence. Coding sequence is called exon and non-coding sequence is called intron. So and exon basically take part in a process of mRNA, while intron do not required in the process of mRNA, and that's why they have to removed. So removal of this intron is called the spliceosomes. So that we will see in the next slide. Okay, so this is about the monocistronic structural gene, which is also called as a split gene. Now why it is called split gene? Because intron is not required, and that's why it have to be removed. So removal is there, that's why it's a split gene. Now another one is called polycistronic structural gene, right? Polycistronic structural gene. And this polycistronic structural gene basically seen in prokaryotic organisms. Okay, so here you can see there is a no split gene. Can you see here? This is a polystronic structural gene, and this is a monostronic structural gene. So in the monostronic, you can see this is exon, intron. Now who is going to tell me that which one is the coding sequence and which one is the non-coding sequence? Yes, I just now told you about in the previous slide that what is coding. Yes, so tell me the response. Give your response fast in the chat box. What is coding sequence? Whether it is exon or intron. Very good, Anmoldeep. Excellent. Exon, right? So exon is what? It is a coding sequence, and intron is non-coding sequence. Especially in the eukaryote, they have to removed during the RNA process, or we can say that during the transcriptions. But you can see in the polycistronic gene there is no any split gene. Can you find any split gene over here? No. See here there is a exon, intron, and that's why it is a split gene. But in case of polycistronic there is no split gene. So it is found in prokaryotic organisms. Now, so we studied what is gene, what is the type of structural gene. Now what are the characteristics of gene? What are the specific characters required to call it as a gene? So first of all, gene, it can be characterized by various methods, but I'm just going to give you 5 important characteristics regarding the gene. The first one is the replications. That means of gene is a unit of genetic material, and if it is genetic material, then it has capable or can able to replicate. If it is genetic material, then it can able to replicate. That means of this is the gene having the capability to replicate their own body. Another one is a recombination. Now again, recombination is what? It is a unit of recombinations that is capable of undergoing crossing over. And you know that when the crossing over take place, there is a recombinations. You will get a different combinations of the organisms. So that means of you required what? You required a crossing over in the recombination process. The third one important characteristics is the mutations. And again, mutation is also a unit of genetic material which undergo mutations, because mutation lead to the variations, right? So mutation is also important, which is a characteristics features of gene. Now the another one, so replication, recombinations and mutations. Now the another one, the fourth characteristics is a somatic structure, or we can say the functions. That means of gene is the unit of heredity, as you know very well, connected with the somatic structure or the functions that lead to the phenotypic expressions. You know very well that how it is connected and how the phenotypic expressions generally observed in the F1 generations, right? That means of gene regulate structure and metabolic activities, metabolisms, so that it will appear the phenotypic expressions. So it basically relate to the somatic structure and functions. And the fifth one, that is development and differentiations. So development and differentiations, you know that gene govern the development and differentiation. So and because of that gene you can able to get a different variations in the organisms. So this is the 5 important characteristics which you need to remember, and that is important for you. Now let's move ahead and try to understand the hypothesis of gene. Yes, hypothesis of gene. Now what is this hypothesis of gene? So we will see that what are the major, you might have heard about the one gene one hypothesis and all. So actually how the gene play the important role for metabolisms or any kind of error or any kind of deformities in the F1 generations that lead to be gene along with their hypothesis. So yes, it was a first time, that is called the Archibald Garrod. That is also, that man also called it as a father of biochemical genetics. Okay, so Archibald Garrod, the father of biochemical genetics, was first to hint that gene operate through the enzyme. That means of he was man who first time said that gene was operating through the enzymes. It was not that means of gene is going to something, some chemical message. No, it is going to be operated through the enzymes, right? It is operated through the enzymes. So he studied a number of inherited human disorders and found that they are inborn error of metabolisms or the failure of metabolic machineries of an organisms to perform a particular functions due to the formation of what? Formation of defective enzymes associated with the inheritance of defective gene, right? So you know that there are various kind of disorders in the human beings, and we have also studied in our pedigree analysis that what type of that is Mendelian disorders and chromosomal disorders. So all these disorders, what it all leads with the certain kind of enzymes. So he studied who? That is Archibald Garrod, who studied a number of inherited human disorders and found that they are inborn error of metabolisms or the failure of metabolic machineries of an organisms to perform a particular functions due to the formation of defective enzymes associated with the inheritance of defective gene. So Garrod studied, alkaptonuria. You know very well that what is alkaptonuria, right? So Garrod studied alkaptonuria, which is a genetic disorders or the disease of human beings characterized by, brown or black color of exposed urine. So he came to the conclusion through this pedigree analysis that the disease was caused by the inheritance of pair of recessive gene, right? So basically actually alkapton is produced in the human beings. Alkapton is what? It is an acid. So this acid is formed through metabolisms of 2 amino acid, and that 2 amino acid is called phenylalanine and the tyrosine. You can see here. So it's a phenylalanine and tyrosine. So these phenylalanine and tyrosines are basically, you know, these are the metabolism. These are basically formed during the metabolism of 2 amino acid. The one is phenylalanine and the another one is the tyrosines. So normally alkapton acid is oxidized to carbon dioxide and water with the help of alkapton oxidase, right? So this is what the normal process. However, in the patient suffering from the alkaptonuria, the enzyme is defective and unable to metabolize this acid, and the result, alkapton acid accumulate in the body, and it shows that gene work through the enzyme. That means of with the help of enzymes we can say with the help of this reaction we can say that the gene basically it operate with the help of enzymes, and that enzyme is called alkapton oxidase. Yes, you can see here it is alkapton oxidase. So oxidase is what? Enzymes. So this 2 amino acid basically it work with the help of enzymes. So he concluded that means of through this it shows that the gene work through the enzymes. Without enzymes gene cannot work, right? So based on this his experiment, the another scientist, that is called the Biddle and Tatum. Biddle and Tatum, the another scientist. Yes, it was Biddle and Tatum in the year of 1948. Okay, in the year of 1948, which state that gene control, what gene controls the structural and the functional trait through controlling the synthesis of specific proteins or the enzyme formed by the latter, right? So it's actually, you know, the Biddle and co-worker, they worked on a different type of observations, and when they found their observation were based on the red eye color of Drosophila melanogaster. Drosophila melanogaster, you know, the organisms, which is a model organisms, right? So this Drosophila melanogaster basically which is controlled by the 2 gene. Their eye color, you know, Drosophila melanogaster eye color is controlled by 2 gene and is caused by the blending of brown and vermilion pigments. That means of 2 gene that carry the 2 traits. One is brown trait and another one is a vermilion pigments. So a piece of larva destined with the to form a vermilion eye can be made to produce a red eye color if it is placed in the body cavity of larva having red eye, because of the later you know, it provides this enzyme for brown color with the transparent lac. So from this conclusion they made that one enzymes, one gene, one enzyme hypothesis. How? Because, and that is proved with the help of Drosophila melanogaster. You know, Drosophila, let me write here example for you. So he worked Biddle and Tatum. Example, that is Drosophila melanogaster. Okay, so this is what, these are the organisms in which he studied the eye color pattern of this Drosophila fruit flies. So he made this hypothesis, that is one gene one enzyme hypothesis. Who made this hypothesis? That is Biddle and Tatum. Now the another one, that is one gene one polypeptide hypothesis. Now what is mean of one gene Okay. Now, one gene, one hypothesis. From that, it is very much clear about the how exactly this correlate. Now, come to the another hypothesis, that is called one gene, one polypeptide hypothesis. So basically, it was Anofsky uh, in the year of 1965 somewhat, that found, the enzymes tryptophan synthase of bacteria E. coli, which consist of two separate polypeptide, and these two separate polypeptide is called polypeptide A and polypeptide B. Now this polypeptide A and B, polypeptide A is of alpha type. Okay, it is of alpha type, and B is of beta types, right? So the synthesis of these two polypeptide is controlled by the different gene, that is called the TRP1, that is tryptophan 1. It is controlled by TRP A, I can write, then TRPB. So it is controlled by more than two enzymes, right? And more than two genes. Now, a change in any of these two genes cause the inactive of tryptophan synthetase through the non-synthesis of alpha or beta polypeptide. So that means of if any one of these gene is going to defected, that means of it is going to be inactivates if there is a defect in any one of the gene. So the synthesis of these two type of polypeptide is controlled by the two different genes situated on the different chromosomes, and therefore one gene, one enzyme hypothesis was changed to one gene, one polypeptide hypothesis. In the year of 1953, it was changed and converted into by Nigrims. It was converted. So the hypothesis stated that structural gene specify the synthesis of single polypeptides. Right? So this is about one gene, one polypeptide hypothesis. Okay. Now this is all about the gene. Now let's talk about the transcriptions. Now transcriptions means what? Now you know the steps of central dogma of molecular biology. DNA, converted into mRNA, and then followed by protein, right? So this is what called, what is this? What is A? What is B? Very good. A is this question mark is replication. A stand for transcriptions, and B stand for translation. Yes. So we will focus on this stage, that is from DNA to RNA. So the conversion of DNA into mRNA is called transcriptions. So a process of copying genetic information from a template strand of DNA into RNA is called transcriptions, and it is mediated by the RNA polymerase. That means of with the help of RNA polymerase, it is going to be. So transcription take place in the nucleus of eukaryotic cell. Well, now you know the basic steps of DNA, right? So this entire, before we get into the details steps of transcription, let's first talk about how RNA polymerase, because that is very important, because as the statement said over here, with the help of polymerase, or we can say it is mediated by the RNA polymerase. So RNA polymerase. So what is this RNA polymerase? How this RNA polymerase take place? The major enzyme involved in the transcriptions. And also we will see that uses of double stranded DNA as a template to make a single stranded mRNA. So now as you know that DNA is a double stranded molecule which is wrapped tightly in a helical shape, as you can see here. Yes, this is what the DNA strand. Yes or no? This is what the DNA strand. Yes or no? Respond it. Is this a DNA strand? Okay. So as I told you that DNA is what? DNA is a double stranded molecule. Yes or no? So for this is the DNA. DNA double stranded, right? And it is wrapped with tightly with the helical shape. As you can see, this is the shape is helical. Yes or no? Helical shaped, right? So RNA polymerase can bind to DNA or unwind the helix to separate the two strands and expose the individual nucleotide. How? So you can see this is the RNA polymerase, and when this RNA polymerase bind to this strand, this two strand double stranded tightly helical shaped structure is going to unwind. So this is what unwind DNA. Yes or no? This is what unwind DNA. Yes or no? Come on, respond it. Yes. Okay. So this unwind DNA, you can see in more. Now here, this is the same of the above one, but slightly labeled, so it will be more easy to you remember this. This is similar diagram, but is slightly different. That is along with the naming. So what exactly here? So you can see here in this diagram, since there are two strands of DNA, which strand is actually being used as a template. That is important, right? To know which strand is used as a template. So one strand is called the sense strand. Can you able to see the sense strand? Yes. Here is the sense strand. Yes or no? See at the base there is a sense strand. Okay. And this sense strand is read in the direction of, so can you read the direction? What is the direction? In the 5' to 3' direction. Yes, this is 5' and this is 3'. So it is read through the 5' to 3' directions. Okay, and correspond to the mRNA that is translated. Now opposite to this, antisense strand is 3' to 5'. So you can see here it is read from the 3' to 5' strand. Clear? And this strand is called the antisense strand, that is complementary to this and is actually the strand used as a template for transcriptions. That means of this is the complementary strand which is used for transcription. So that means of this antisense strand is used as a template strand. Is this clear? Yes. So out of this DNA strand, one strand is called sense strand, another strand is called antisense strand. Sense strand is read in the direction of 5' to 3', whereas antisense strand read from 3' to 5' strand. and this antisense strand is considered as a what? As a, actually it is a template for the transcriptions. So you can, as you can see here in the diagrams. Is this clear? Okay. Now here you can see one important things, and that is what? The RNA transcript. Yes, RNA transcript. Now you can see this RNA transcript, the strand of this RNA, you can see there is uracil. Yes or no? Uracil is there. Yes or no? Yes. So that means of in this particular diagram, when the RNA is transcribed, thymine is replaced by uracil, and this indicate that DNA has a thymine is replaced by the RNA base, that is called the uracil, right? So that's why there you can see there is no any kind of, can you able to find any thymine in this strand from here to here? Can you able to find the T in this complete sequence? No, right? That means of in the DNA there is a thymine which is going to be replaced in RNA and become a uracil. This is uracil, right? And this is thiamine. Thiamine. Clear? That means of that you have to remember. Yes, that what is replaced in RNA. RNA means what? It is a complete ribonucleic acid. It is replaced by that thymine is replaced by uracil. That means of no thymine present in that sequence. So if any sequence given to you, you have to identify whether it is DNA or RNA. So how you will identify? With the help of this nitrogen bases, with the help of these 4 nitrogen bases, that is adenine, guanine, thymine, cytosine, uracil, right? So if in the strand, if there is uracil, what that strand called? If suppose there is one strand having all the 4 amino acid including uracil, if uracil is present, then what kind of this strand is called? This strand is called RNA, right? And if uracil is absent, that strand is called DNA. It's very simple for you to remember. Yes or no? Okay. Let me write one particular strand yes, I'm writing one strand and you have to tell me whether it is DNA strand or RNA strand. I'm not going to ask you. I'm going to ask with the help of calling names. So once I call, then only you respond. No one can respond in the chat box. Okay, now this is one single strand and having uh, a okay, this is considered as a strand and so on. So tell me what kind of strand is this? I want answer from Prasanthi. Prasanthi, are you there? Yes. Okay. So please respond it. What kind of strand is this? No idea. Prasanthi responded fast. What kind of strand is this? Is it RNA or DNA? Sasidharan? Sasidharan responded fast. Okay, Prasanthi said RNA. Sasidharan said RNA. Okay. So yes, this is RNA strand. Why it is RNA? Because it is thymine is replaced by uracil, and that's why in this strand there is no thymine, no T is present in this strand, and that's why it is what? T is absent in this strand, so it is called RNA strand. Okay, so now it is very much clear about that. Once the DNA transcript, it converted into RNA, which lost the thymine and converted into uracil. Now, so this is about the strand, antisense strand and sense strand. Now we have understood that how RNA polymerase use DNA as a template to form the mRNA. Now let's go the process of actual steps of transcriptions. So before that, there is a requirement of transcription unit. Now what is the transcription unit? Let's see about it. The transcription unit. Now here transcription unit in the DNA molecule that basically differentiated into three regions, and that region is called, the first one is a promoter region, okay, promoter, structural gene and the terminator gene. Three important region is there in the transcription unit. This is a transcription unit. Okay, that means for transcriptions, what are the important steps is, or I mean to say requirement is there. So if there is a transcription unit, it should be a promoter, there should be a structural gene, and there should be a promoter. Okay, now what is this? Let's talk about what is structural gene, promoter. Okay, so yes, promoter, it is the region, you can see here in the diagram also, it is the region on DNA molecule to which RNA polymerase bind and initiate the transcriptions. Okay, that means of RNA polymerase is going to bind on this strand. Okay, so this is a coding strand, and RNA polymerase, this is, suppose this is RNA polymerase, the yellow box is a RNA polymerase. So RNA polymerase is going to bind at the promoter side. Okay, so what is promoter? It is the region on DNA molecule. Okay, it is a region on the DNA molecule to which RNA polymerase bind and initiate the transcriptions. That means of actual initiation of transcription started from the promoter. And promoter is located, where is promoter located? So it is always located on the upstream towards the left of the structural gene. Now you can see where is the structural gene. This is the structural gene. Yes, so it is always present towards the left side of the structural gene. That is what the promoter region. Clear? Yes, so it is always present at the left side. Now the another thing, that left side means towards the 5' end of coding strand and 3' end of template strand. So you can see this is the template strand and this is the coding strand. So in the coding strand, it is lies towards the 5' end. At the template strand, it is lies towards the 3' end. This is what the promoter. Yes. Now in most eukaryotic organisms, okay, in most of the eukaryotic organisms, the promoter has AT rich region. Okay, in most of eukaryotes, promoter has AT rich. What is mean of AT rich? That means of there is a, in the particular sequence there is a n number of 80, 80, 80, 80 is there, right? So they have a number of it has a 80 rich region, and because of this 80 rich region, it is called Tata box. TATA Tata box. Clear? That means of if there is a promoter, suppose this is a promoter region, so on this promoter region there is a continuously AT GC AT GC AT. That means of 80 80 80, that means of 80 number is several times, no? So because of this strand is called 80 rich strand, and if it is 80 rich, then it is referred as a Tata box. Clear? It is referred as a Tata box. Now the area has a groove to which a specific protein component can combine Tata box, and when in this groove Tata box is going to combine with the particular proteins, and that is called the now box. That is called the now box. This is W. So there is a now box. Clear? So Tata box and now box, you should be very clear with these two box. Okay. What is Tata box? It's a very simple. The promoter has 80 rich region, that is called the Tata box, and the area has a groove to which the specific protein component can combine Tata box. That means of this is going to combine, means I can draw a figure like this. Suppose this is the promoter having 80 rich, so it is called the Tata box. Tata box, and this Tata box is going to attach with another box which is called the Pribnow box. Pribnow box. Okay, so it is Pribnow box is a kind of, we can say grooves to which the specific protein component can combine Tata box. It is called the Pribnow box. Now why it is called Pribnow box? Because it's discovered by the Pribnow. So Pribnow is the name of the scientist, and that's why it is a Pribnow box. Pribnow in the year of 1975, and that's why the name is a Pribnow box. So Pribnow box was discovered by Pribnow. Okay, and so this is Pribnow box in prokaryote. Clear? Pribnow box in prokaryote. Similarly, there is a Hogness box. Hogness box. So this is in case of prokaryote. Sir, can you repeat the meaning of Pribnow box? Yes, see here, a Pribnow box is what, Achilles, let's try to understand. So basically, when we're talking about the Tata box, you are very much clear about the Tata box. What is Tata box? That is a promoter has 80 rich site, rich region, that is called Tata box, right? So in that 80, in the Tata box, the area has a groove to which a specific protein component can combine Tata box. So when the specific protein component combine with the Tata box in prokaryotic organism, it is called Pribnow box. Why Pribnow box? Because it's after its discoverer name, that is called Pribnow in the 1975 in the prokaryote, and that's why it is a Pribnow box. But in eukaryotic organisms, it is referred as a Hogness box. Okay, in eukaryotes, Hogness box. Okay, so it is same the name, that is the called the Hogness. Discoverer is Hogness, so it is a Hogness box in the eukaryotic organisms. So this is in eukaryotic organisms and this is a prokaryotic organisms. Is this clear? If Tata box combined with the Pribno box, that is prokaryote. Hello? Yes.
So, like that organization is same as but
Yes, pardon?
so like Pribnovox Hackness is other name of Pribnovox, but Hogness in prokaryotes, but Pribnos in eukaryotes.
No. Let me tell you. See, I think so something is from, okay. See, Pribno box, when suppose there is a prokaryotic organisms, so in the prokaryotic organism there is a Tata box, and in the prokaryotic organisms once it joined a Pribno box, it is called the Pribno box, right? That means of there is a proteinaceous component when specific protein is going to bind, that is called the Pribno box. Okay, but in case of you, suppose that organism is eukaryote. So in the eukaryote, if there is a specific protein when it bind with the Tata box, it is called the Hogness box. Means it is basically similarly same, but it depends on organisms to organisms. If it is prokaryote, it is called Pribno box. If it is eukaryote, it is called the Hogness box.
Okay.
Clear? Okay. Yes, Achilles, is this clear to you? Okay. So, okay, apart from this promoter, in eukaryotic organisms, it also required enhancer. Okay, enhancer. Enhancer is also required in eukaryotic organisms, but in prokaryotic organisms, no enhancer is required. So this is about the promoter. Now let's talk about the structural gene. Now, what is structural gene? It is the area of the template strand. Now, what is template strand? You can see this is the template strand. Yes or no? Can you able to see this? Yes, this is a template strand, and here you can see this is structural gene. So what I said, it is the area of template strand that is involved in the transcriptions and formation of RNA. That means of this, a particular area is going to involve in the transcription. This is the transcription site, right? That means the transcription actually start here, where there is a formation of RNA take place at this region, and that's why this is called the structural gene. Okay, so and this is common to all the organisms. So structural gene means what? It is the region, or we can say it is the area of template strand that is involved in the transcription or formation of RNA. Now, structural gene is of two types, as I told you. What this structural gene of two types? One is called polycistronic and the monocistronic. Yes or no? I told you in the beginning of the gene, yes or no? What is polycistronic? What is monocistronic? So monocistronic is seen in eukaryotic organisms. Polycistronic seen in prokaryotic organisms. Right? So monocistronic structural gene carry information for the synthesis of one polypeptide chain, and they are mostly found in the eukaryotes. And polycistronic structural gene carry information for synthesis of more than one polypeptide chain. Okay, so poly and mono, the difference is very clear. Okay, monocistronic means carry one single polypeptide. Yes, it carry one single polypeptide. One polypeptide chain. That is called the monocistronic. In case of polycistronic, So it carries more than two polypeptide chains, so it is called polycistronic. Okay, so that is what the structural gene. Okay, is this clear about the structural gene to all of you? What is structural gene? Very first of all, structural gene is an area of template strand that is involved in the transcription of, or we can say the formation of RNA. Now this structural gene is divided into two types, monocistronic and polycistronic. Monocistronic structural gene carries information for the synthesis of one polypeptide chain, which is generally found in eukaryotic organisms. Similarly, in the polycistronic structural gene, it carries information for synthesis of more than one polypeptide chain, and they're mostly found in the prokaryotic cell. So polycistronic, prokaryotic, monocistronic, eukaryotic, that is what the structural gene. Is this clear about the structural gene? See, I want response from everyone, not just from Achilles and Anmoldeep. Achilles and Anmoldeep responding very nicely, but I don't know what about others. All of you, please respond it in the chat box. Do respond fast. See, Ragul, Sadhana, Prasanthi, then Gopinath, then there is Sasidharan. Yes, number are huge, but no one responding. Why? Only 3 students responded: Achilles, Bhuana, and yes, Anmoldeep. Sasidharan responded. Okay. Okay. Now see the last one, that is called the terminator gene. Okay, terminator. Hope this is clear to all of you. Promoter, structural gene, and now come to the terminator gene. Now what is terminator gene? Now the name itself indicates terminator means what? This region on the DNA molecule is present at the downstream right of the structural gene. Now where is the right and where is the left? So you can see this is the left side where the promoter is present, and the right side there is a terminator is present. So you can see here this is the third. So this is the first one, this is the second one, and this is the third one. That is called the terminator. Now, so what is the location? It is present at the right side, or we can say the downstream. Okay, this is something important. This is called the downstream. Let me write here. This is called the downstream, and this is called the upstream. Upstream. Clear? Downstream and upstream. Is this clear? Yes. Upstream and downstream. Okay. Now, so as I told you that terminator is present in the downstream right to the right of the structural gene towards 3' end of coding strand and 5' end of template strand. So you can see here this is 3' of template and 5' of template and 3' of coding strand. Now this terminator usually define the end of the process of transcription. Termination means what? That means it is going to terminate, or we can say the transcription process is going to be end. So it is the end of the transcription process. So terminator means going to end the transcription process, end of transcription process. That is what the terminator. So this is what the transcription unit which is required for the transcriptions. So this region you should know very well, that what is promoter, what is structural gene, what is terminator gene, what is template strand, and what is coding strand. Okay, let me confirm that. Is this clear to everyone about the actual structure of transcription unit? Or anyone have a doubt, then please ask me again. I want response from everyone. Anyone have a doubt, please respond it fast in the chat box. Come on. Okay, Achilles said no doubt. Okay. Okay, so only one student, no doubt. What about others? They all having doubt? So only two responded. Achilles and Prasanti. No doubt and clear. What about others? Okay, Sasidharan, please respond it. Okay. Well, let's move with the next. So this is the complete structure. As you know very well, I'm not going to tell you this again. Now you have to respond. So here you can see this is a promoter which is present at the upstream gene, and this is terminator which is present at the downstream, and this is called the gene which is called structural, or we can say the coding region. This is structural gene, or we can say the coding gene, or right. And so these are the transcription unit. Yes. Now transcription unit, it also required some material. Like suppose there is a experiment and we required a lot of material to perform that experiment. Similarly, for transcriptions, you know, they required a particular what we can say the materials for the transcriptions. So what are the important material required for the transcriptions? Very first, that it required enzymes RNA polymerase, and you know that RNA polymerase is going to bind at which region? Just tell me. This RNA polymerase is going to bind with the promoter or structural gene, where or the RNA polymerase is going to bind. Promoter. Very excellent. Excellent answer. It is going to bind with the promoter for initiations of the transcriptions. Very good. Then it required DNA template. 4 type of ribonucleotide triphosphate. You know that ribonucleotide triphosphate, adenosine triphosphate, CTP, thymine triphosphates, cytosine triphosphate, G for guanine triphosphate, and UTP. Why UTP? Because it is a RNA sequence, ribonucleotide sequence, right? So it is a UTP because thymine is replaced by uracil. And divalent metal is ion is also required, and that divalent metal ion is magnesium or manganese, Mn, as a cofactor. So these two, Mg and Mn plus 2, that are required as a cofactor for as a transcription unit. No primer is needed for the RNA synthesis. That means for synthesis of RNA, there is no requirement of any primer. Okay, means for RNA primer is not no any single primer is required for RNA synthesis. So this is the unit of transcriptions. Now let's try to understand the actual process. How this DNA converted into RNA, or we can say mRNA. So first we will see in prokaryote, and then we will see in eukaryote. Okay, so prokaryote means basically in bacteria, all the lower organisms, right? So in bacteria, synthesis of all type of RNA are catalyzed by a single RNA polymerase. That means with the help of single RNA polymerase, this entire transcriptions take place. So transcriptions in prokaryote is divided into 3 important steps, and that steps is called initiations, elongations, and termination. Initiations, elongations, and termination. Now, what is happening in initiation in the prokaryote? So you can see here the initiations is started with the in prokaryote initiations it occurs in the, that means the transcription basically in prokaryote, transcription it basically take place in the cytoplasm with the help of DNA dependent RNA polymerase. Okay, it is DNA dependent RNA polymerase. It take place in cytoplasm of eukaryotic cell. You know the cytoplasm? See, you have already studied the structure of cell in your lower grade, right? So you should know that what is a cytoplasm, right? So in prokaryotic cell, this transcription take place in the cytoplasm, but in eukaryotic cell, it take place in the nucleus. That is the major difference between the prokaryote and eukaryotic transcription. Okay, so it take place in the cytoplasm. Second things, with the help of DNA dependent RNA polymerase. It is a type of DNA dependent RNA polymerase. Clear? It's a DNA dependent RNA polymerase. Another one. So this catalyze all the three type of RNA. All the three type of RNA means it is going to catalyze all the three type of RNA. That means of, you know that RNA is of three types, and these three types is stand for rRNA, then mRNA, and then tRNA, right? So it is going to form this all three type of RNAs in the bacteria. So all these three RNAs are needed to synthesize a protein in the cell. Means all these three type of RNA, rRNA, mRNA and tRNA, all three type of RNA is required for what? Required for protein synthesis in the cell. It required for protein synthesis in cell. Clear? So the complete process of transcription in total 3 to 5 steps. Now let's try to understand the initiation stage means what. Now in the initiations, RNA polymerase reach the promoter region. Can you see here in this diagram only itself, this is the RNA polymerase. Yes or no? And this is what the DNA double helix. So this is the DNA double helix and this is the RNA polymerase, and in this particular DNA there is a promoter, there is a structural gene and there is a terminator gene. Everything is present in this particular sequence of DNA double helix. So what happened? The RNA polymerase, it reached to the promoter. So once it reached to the promoter region, what happened? So RNA polymerase has a sigma factor, means RNA polymerase itself having a sigma factor, and because of this sigma factor, okay, this having a sigma factor, so RNA polymerase initiate the transcriptions with the help of these recognizations. So it use nucleoside triphosphate as a substrate and polymerase in the template dependent fashions following the rule of complementary, and it also facilitates the opening of helix. So as soon as it bind, it start facilitating as a helix. So you can see the initiations. So you can see here how it started. So they have initiation factors and it will start. So this is the transcription direction from 5' to 3' directions. Okay, so you can see this cause the local unwinding of the DNA double helix. That means of once the promoter site, RNA polymerase bind at the promoter site of the DNA, what happened? This cause the local unwinding. Local unwinding means DNA is a double helix like this, but because of the DNA polymerase, it is going to unwind like this, right? So it is cause the local unwinding of the DNA double helix, and as they have, as this RNA polymerase is having the initiation factor which is called the sigma factor. Sigma factor is called initiation factor, and because of this initiation factor it is start initiating the RNA synthesis. Means this is the first steps of the initiations, and that's why it is a initiation factors. Clear? So what is important in this complete process in initiations? RNA polymerase, it going to recognize, it going to bind with the promoter, and this RNA polymerase having a sigma factor, and this sigma factor represent as an initiation factor. Okay, and because this initiation factor present in the RNA polymerase initiate the RNA synthesis. Here, and once it starts in its synthesis, that means of synthesis of RNA, there is a winding of DNA double helix take place. So double DNA helix is there unwinding, and because of this unwinding RNA synthesis take place. So here you can see this is unwinding, and then it starts synthesis of RNA. Clear? So this is what the initiation factor. Initiations. Is this clear to everyone? That what is the transcription in prokaryote? It is initiations. Yes. Anyone have a doubt? So this is important. Huh? This is important, which I have written over here. What is I mean to say, if someone asks you that where the transcription in prokaryotic take place, it is in cytoplasm. It is what type of polymerase? So it is a DNA dependent RNA polymerase. So it synthesize all three type of RNA, that is RNA, mRNA and tRNA, and all these three tRNA are for protein synthesis in the cell required in the prokaryote. Clear? So this is about the initiation. Now let's talk about the next one, elongation. Now what is mean of elongations? So you can see here, elongation, the RNA polymerase, which is also referred as a core enzyme, in the presence of energy and magnesium, can catalyze the process of elongation. So you can see here you can see this is the elongation. So after initiation, the next step is elongations. So in the elongations, you can see that RNA polymerase in the presence of enzymes, means this is the RNA polymerase along with the enzymes, or we can say some amount of energy plus some metal, and that metal is Mg plus 2. Mg plus 2. Okay, so this metal, kind of element, we can say, not metal, it is a kind of element. So what happened? Together they catalyze the process of elongations. So as the enzyme move along the DNA template, RNA chain become longer. So you can see here, initially the RNA chain is very short, now it become going to be enlarged. So this yellow color is what? This is the RNA chain. Okay, this yellow color is nothing but it is called the this band thread like structure is called RNA chain. So this RNA chain is going to be elongated because of this elongation process, right? So in this process, activated ribonucleotide triphosphate are added. It is a complementary to the base sequence of the DNA, and this complete RNA chain is going to be elongated, and that's why its name as a synthesize in the direction of 5' to 3' directions. It is always from 5' direction to 3' directions. Clear? Yes, this is what the elongation steps. Anyone have a doubt in this elongation steps? Okay. Now come to the next step, that is called the termination. Now a termination factor. Now you can see there is a requirement of termination factor, and that is called the rho factor. This signature is represent the rho factor. Okay, so rho factor bind to the RNA polymerase, means RNA polymerase, when there is a rho factor bind with this RNA polymerase, it terminates the complete transcriptions. And it once it reach to the terminator, there is a row factor, and this row factor when bind with the terminator, I mean to say polymerase, RNA polymerase, it stop the complete process. So it's a terminations. So what happened, you can see RNA synthesis take place. This is the complete RNA synthesized. Now here this RNA and polymerase row factor is going to be separated, and this reaction go ahead. That means of it stop the complete reactions. Now RNA is prepared. Now this RNA reach into the site for the protein synthesis. This is how the actual process take place in the prokaryotic cell. Clear? So you have to remember RNA in prokaryote synthesis of, that means of transcription is very simple in the prokaryotic cell, right? So in bacteria, mRNA does not require any processing to become active. So transcriptions and translation take place in the same compartment. Transcriptions and translation take place in the same compartment, as there is a no separation of cytosol and the nucleus, right, in the prokaryote. That means of in the prokaryote, transcriptions and translation both are take place. Many times the translation can begin much before the mRNA is fully transcribed, and thus the transcription and translation can be coupled, right? That is the case of transcription in prokaryote. Yes, anyone have a doubt in this transcription in prokaryote? Okay. So here you can see in the bacteria, transcription and translation can be coupled because mRNA require no processing to become active, and transcription and translation take place in the same compartment. You know why? Because there is no separation of cytosol and the nucleus in the prokaryote. That is the reason behind. Now come to the transcription in eukaryotes. Okay, now we studied transcription in prokaryote. Now eukaryote. Eukaryotic transcription in eukaryote is little bit more complex. It is more complex as compared to the prokaryote, because prokaryote is very simple process, right? Like there is a binding of RNA polymerase, sigma factor start initiating the reactions, transcriptions, and then there is a elongation of RNA, and then followed by once it reaches to the terminator, there is a termination row factors who terminate the complete reaction. This is very simple in the prokaryote, but in the eukaryote it is very complex process, and it having a totally different structures, and that's why in the eukaryote there are two additional complexities are there in case of eukaryote, and these are 3 RNA polymerase. That means of in case of prokaryote there is only one polymerase is enough, but in case of eukaryote there is a two additional complexity. One, that is 3 RNA polymerase is required, and there is a primary transcript contain exon and intron that are non-functional, hence intron must be removed. So that means of these two process is taking place in the eukaryote, and then only transcription is possible. So let's try to understand what are these two important additional complex cities in the eukaryote. So very first, the 3 RNA polymerase in the eukaryote is RNA polymerase first, second, and third. Okay, it is first, second, and third. What is RNA polymerase first? It transcribes the rRNAs, means 20S RNAs, 28S rRNAs, like this. Okay, 18S and 5.8S. You have to remember, this is important. All these 3 type of RNA along with their subunits is very important, that you have to remember. Okay, so 3 type of RNA polymerase in eukaryotic, and these are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Now in case of, so this basically transcribe only rRNA. Okay, that is RNA polymerase 1 transcribe only rRNA. Now RNA polymerase 2, it transcribes the heterogeneous nuclear RNA, that is called hnRNA, and it is the precursor of mRNA. Means basically RNA polymerase 2 is associated with the mRNA, and RNA polymerase 3 transcribe the tRNA, 5S RNAs, snRNAs, that is called the small nuclear RNAs. So this is stand for tRNA. Okay, so that means of all three RNAs is required for transcriptions in the eukaryotes. Is this clear? Okay. Now another thing, that in eukaryote transcriptional unit has only one gene, right? As I said, monocistronic, right? Monocistronic is found in eukaryotic organisms, polycistronic found in prokaryotic organisms. So we are studying eukaryotic organisms, so it has only one gene. So initiation of transcriptions require protein, and that is called the transcription factors. So basically here sigma factor is absent. As we have seen that in prokaryote there is a sigma factor, yes or no? In prokaryote there is sigma factor, yes or no? Yes. So in case of eukaryote there is a no sigma factor, means in this particular eukaryote there is a no sigma factor. That means of sigma factor absent. In what? In the eukaryote. Okay. No primer is required to start the reactions. Here no primer required. No primer required. Okay. Then RNA are released and processed in the nucleus. Means in a eukaryote, RNA released in nucleus and process in the nucleus. Okay. And coupled transcription translation is not possible in the eukaryote. So greater part of the product pass from the nucleus into the cytoplasm. Means here in case of prokaryote there is a cell inside the nucleus. So transcription take place in the nucleus and it transported to the cytoplasm where the translation take place. This is the case of translation take place. This is the case of eukaryotic cell, right? But in prokaryotic, transcription and translation both are take place in the cytoplasm. Are you getting my point? Okay. So the primary transcript is converted into, now here one more important thing, that is the newly formed RNA synthesis RNA polymerase 2 is called the hnRNA, that is a heterogeneous RNAs, and the primary transcript contain both unwanted base sequence alternate with the useful base sequence, that is called exon and intron. So let's try to learn the actual process. Now here transcription in eukaryote. So it start with the splicing. Now what is splicing? So you can see it is similar to the sequence that is from the initiations, elongations and transcriptions, and after the RNA synthesized, this is the newly synthesized RNA. Okay, this is the newly synthesized RNA. You can see here, this is newly synthesized RNA, okay, in the eukaryote. So this is what the post transcriptional modifications, that what happened after the newly formed mRNA or RNA in the eukaryote. So first it undergo a splicings. What is mean of splicings? So from hnRNA, what is hnRNA? hnRNA is those type of newly synthesized RNA which having both exon as well as intron. Exon is a useful base sequence which is called the coding sequence, and intron which is called unwanted base sequence which is called intron, right? So it is not required, so it is a non-coding sequence, right? So what happened from the RNAs, introns are removed with the help of splicings. What happened? Introns are released. So introns if released and exons are spliced together, that process is called splicing. So you can see here, this is the black band is called the intron. So this intron has to be removed from this exon. So you can see how it coiling and it form a circle, and it later it get removed from this particular strand, right? So this is what that removing of this intron is called splicings. And capping, so you can see capping here, the nucleotide methyl guanosine triphosphate cap is added to the 5' end of RHNRN. So this is called 5' methyl guanosine PPP. So this is going to be that is called the capping. And tailing, that means of polyadenylations, means here adenylated residues are added at 5' end. So you can see here the poly tail added at the 5' end. So this is what the splicing, capping and tailing. This is three important process after the post transcriptions. Okay, that means of once the transcription done, then these transcriptions having the HNRNAs having the intron and exon. So intron have to be removed. That process called spliceosomes, and exons are sliced together. It form a transcriptions. So this is what the complete process. So, and this is what called the processing of primary transcription. So you can see this is a exon, intron, exon, intron, exon. So we have to remove the intron. So with the help of RNA processing, what happened? This exon is going to be removed. So that process is called the intron. And here you can see at the 3' end there is a poly A tail addition of poly A tail. At the 5' end there is a 5 capping is there take place. Now, so there is a RNA synthesis. That means of RNA is formed, and that RNA is either in the form of mRNA, rRNA or tRNA, right? So you can see here the type of RNA. Yes. Okay. So what are the type of RNA? So there are total 3 type of RNA, as I told you in the beginning of the process of transcriptions. Now how? So you can see here rRNA stand for ribosomal ribonucleic acid, right? That is called rRNA. So it is the most abundant RNA which has 3 to 5 types, and some of its type are the longest of all RNA. So what are these type? 20S RNA, 23S RNA. These are the types of rRNA. Okay, as the name indicate, RNA is a constituent of the ribosomes, because rRNA, so it stands for ribosomes, right? So here it lies coiled in between and over the protein's molecule. So you can see the coiled structure, base paired, and so this is a coiled region. That is what the rRNA. Now, what is the function of this RNA? So basically, as the name itself, ribosomal RNA. So RNA bind protein molecules and give rise to a ribosomes, right? So 3' end of 18S RNAs, 16S in prokaryote has a nucleotide complementary of those cap region of mRNA. So this is, you can see this is ribosomes, rRNA inside, this is the ribosomes. So ribosomes, then there is another, then this is the 5S RNAs occurred surrounding the complex providing binding site of tRNA, and RNA get associated with the specific protein to form a ribosome subunits. Now what is this ribosome subunit? So you can see in the prokaryotic organism, ribosome is 70S. Why it is 70S? Because when it is not bind, then it is a 50S subunits, which is called the large subunits, and this is the 30th subunit, which is called the small subunits. So large and small subunits are there, and their range is 50S and 30S and 50S. And when this 30S and 50S bind together, it form a 70S ribosomes. and that is specifically found in ribosome, prokaryotic organisms. Similarly, in eukaryotic organisms, you can see it is also having a large subunit and the small subunit, but the number is vary. So number is large subunit number is that is called the 60s, and the small subunit is called the 40s. And when this 40s and 60s combine, it form a 80s ribosomes. So always keep in mind 70s ribosomes found in prokaryote and 80s ribosome found in eukaryote. What are the subunits of the 70s? There is a 50s and the 30s. About the eukaryote, it is a 60s and 40s. That you have to keep in mind, which is important for you. Okay. Now the next one. Now the structure of RNA, tRNA. Now the next type is, this is the tRNA. So first rRNA, now this is tRNA. Now what is tRNA? So let's focus on the tRNA. So as you can see here, tRNA, that is called the transfer RNA. It is also called an adaptive molecule or adapter molecule. It is also called the adapter molecule. Okay, always keep in mind, it is also called the adapter molecule. Adapter molecule. Okay, or you can call it as a transfer RNA. Transfer RNA. Transfer RNA. Now here, transfer RNA, basically it is called the soluble. It is also a soluble or sRNA, in which form it is known before the discovery of genetic code. So there are over 100 type of tRNAs, and tRNA can constitute about 15% total RNA, means it is 15% of the total RNA. What about rRNA? rRNA constitute about 80 to 90%, and now this is 15% rRNA. Clear? Now the nitrogen base of this several of its nucleotides get modified, so it become a pseudouridine, dihydrouridine, DHU, then inosine, ribothiamidine, and all which is there. Cleaver leaf model, we can say cleaver leaf form, and even there is another form. But here let's try to understand the actual structure of the tRNA, which is very important. Now you can see the structure of tRNA. Yes. So you can see the structure of tRNA. This is the model which is called the cleaver leaf model. Okay, Hawley's two-dimensional cleaver leaf model of tRNA, transfer RNA. Now what this model represent? So you can see in this model, 5 regions are unpaired or single stranded. That is A binding site, then T site, C loop. So this is A binding site. Yes. Then this is a T loop, or you can say T site C loop. Okay, this is a T loop over here. Then there is a DHU link. DHU, that is called the dihydrouridine. This is called DHU loop. This is a T loop. This is a DHU loop. Then there is extra arms. So you can see the extra arms. This is the variable loop. Okay, variable loop means this one is the variable loop. This one is a variable loop. Then there is an anticodon stem, anticodon loop. This one is an anticodon loop, right? So you just name it. This is 2, this is 3, this is 4, this is 5, and the one is, I said that is called acceptor stem or amino acid attaching strand. So total 5 regions of unpaired single-stranded AA binding site. Yes, AA binding. What is AA binding? That is called the amino acid binding site. So this is amino acid binding site over here. So this is the first one. Second is T loop. 3 is D loop, or we can say DHU loop. Then there is a variable loop, and then there is a anticodon loop. This 5 region is very important in the tRNA structures. Now let's try to understand what is all these loops. So as I said, the anticodon loop. Where is the anticodon loop? You can see at the base. This is the anticodon loop, right? So this anticodon loop, it is the amino acid binding. It has 7 bases, out of which 3 base form a anticodon. So you can see at the base only 3 base, total 7 is there, right? 1 2 3 4 5 6 7, total 7 amino acids are there, but only 3 at the base which represent themselves as an anticodon, only 3, and that 3 is recognized and attaching the codon of mRNA. Then come to the opposite of this, that is called the amino acid binding site. This is the another one, amino acid binding site. Now here in this amino acid binding site, what is this? It is the amino acid binding site, and its site lies at the 3' end opposite to the anticodon and has a CCA-OH group. Can you see here? CCA, and this is OH group. So it has a group that is called CCA. It attached with the 3' end. Clear? And this is called the recognition site. Okay, this is called the recognition site. This is recognition site. Now T psi C loop, it has 7 base, out of which psi, that means of called the pseudouridine, and RT, that is a ribothymidine, are unusual bases. So you can see these two are unusual bases. Can you see the red one, T and psi? This symbol is called it as psi. So this is thymidine and pseudouridine, which is unusual bases. Okay, in the T psi C loop. Clear? Then come to the next, that is called the DHU loop. So this loop contains 8 to 15 bases, and it has largest loop and has a diuridine. So it binds site for the amino acyl synthetase enzymes. And the last one, they have a extra arms. So this is what the total arms which is represented, and each arms have a important functions. So this model is referred as, very commonly it is called the clover leaf model of tRNA. Clover leaf model of tRNA, transfer RNA. And this is important, so please remember this important structure: DHU, then T psi C, then binding amino acid attaching site, anticodon site, and variable loops also. So as I mentioned over here, it is also called adapter molecule because it's going to adopt that particular sequence. So it is adapter molecule. Now for the initiation, there is another tRNA called the initiator tRNA, and there are no tRNA for the stop codon. Secondary structures of tRNA look like as a clover leaf, and that's why the model, which we can call it as a clover leaf model. Now what is the functions? It is the first postulated by the Crick. So tRNA is adapter molecule. There are different tRNA for different amino acid. tRNA plays a very specific amino acid at a particular point during the polypeptide synthesis as per the codon of mRNA, and codons are recognized by anticodons of tRNA. So specific amino acid are recognized by a particular activating, or we can say amino acyl synthetase enzymes. Clear? So these are the functions 4 or 5 functions which is important. So you just have to remember the important functions for tRNA. Now the last one is a mRNA, which is called the messenger RNA. So messenger, at the name itself indicate messenger. So basically it carry all the informations like so it is a long RNA which constitute about just 2 to 5% of total RNA content of the cell. Means out of total 100, this is only 2 to 5%. Okay. Now it bring the instructions, or we can say information, or we can say the message from the DNA for the formation of particular type of polypeptides. So mRNA is therefore called as an informational, or we can say genetic RNA. It is also called it as a messenger RNA or informational RNA or genetic information. Okay. And that genetic informations, the instructions are present in the base sequence of its nucleotide, and that nucleotide is called genetic code. Clear? So that information which is carried by the messenger RNA or mRNA is called, that code is nucleotide, and that nucleotide is called the genetic code. So 3 adjacent nitrogen base specify a particular amino acid, and therefore it is called the messenger RNA. Clear? Okay. Now here you can see the cap, initiation codon, which is non-coding region, leading regions, and this is termination region, non-coding region, coding regions, and this is poly-A tail. So this is how the region actually represent over here, and each region having a particular we can say functions, right? Like this formation of complete, they are called the polycystonic, while the later it is known as a monocystonic and polycystonic and all. So that we have already studied. What is the major function of this mRNA? So the basic function is mRNA carries coded information for translation into a polypeptide formations. Through the reserved transcriptions, it can form a compact gene which are used in a genetic engineering. Okay, and the phenomenon also occurs in the nature and has added certain gene in the genomes. It has a cap region for the attachment of ribosomes. Cap protect the mRNA from the degradation of enzymes, and mRNA has a tail region for the protection from the cellular enzymes and detached from the ribosomes. So these are the basic functions performed by the mRNA. Now these are few questions related to the today's concept, that which is adapter molecule among the following. I think so this is very simple questions. Come on, do respond fast. Which one is the adapter molecule among the following? Yes, it's very simple, tRNA. Next, which of the following is the most abundant animal cells? RNA in the most abundant animal cells. Yes, it's RNA. The RNA percent is 80 to 90%, followed by tRNA, which is 15%, followed by mRNA, which is 2 to 5%. So most abundant is what? rRNA. Next, methyl guanosine triphosphate is added to the 5' end of the hnRNA in the process of what? Splicing, capping, tailings, or cuttings. I know this is also very simple question. Tell me fast. See itself, see what is saying here, methyl guanosine triphosphate at the 5' end. So 5' mg, you might have seen that in the process of transcriptions in eukaryote, and that is what called capping, right? Tailing means what? There is a poly A tail. poly A tail attached, that is called tailings. Capping means capping of 5 mg, that is called the methyl guanine triphosphate. Splicing, removal of introns. So it's a B answer. So this is what we studied today about the gene. Then we also studied about the antisense and directional transcriptions, that how it transcribe and convert into mRNA, like here you can see. And we also studied about the messenger RNA, tRNA and the ribosomal RNA.